Before we go into the details of the visual pathway, we wanted to give a sagittal view of the eye to show the basic anatomy of it. So a light stimulus is going to come in through the cornea and the lens, and those are going to help focus the light to the retina, which is filled with photoreceptor cells of cones and rods, and in the fovea is a very dense location of cones. Okay. So the light will hit a photoreceptor cell, in this case it's a cone in the retina, then it will go to a bipolar cell in the inner nuclear layer of the retina, then it will go to a ganglion cell in the retina, and that is where partial decussation of the optic chiasm occurs, and that's when the optic nerve fibers on the medial sides of each retina cross over to the opposite side of the body midline. And then the signal will go to the LGN and the thalamus, which is the dorsolateral geniculate nucleus, and then it will go to the primary visual cortex, which is V1 to V2. When talking about partial decussation, it's important to note that half of the neurons will stay on the same side, which means they are ipsilateral, and the other half will cross the hemisphere, which means they are contralateral. This happens at the optic chiasm. In the dark, photoreceptor cells tonically release glutamate. And then the on-bipolar cells have metabotropic and G protein coupled receptors, and they respond to glutamate by hyperpolarizing, which means that the retinal ganglion cell will be inactive. When exposed to light, photoreceptor cells decrease glutamate release. On-bipolar cells respond to the lower glutamate release by depolarizing the cell. This will cause an action potential to occur in the retinal ganglion cell. The pathway that happens, or that moves to, is the thalamus, and then it goes to the primary visual cortex. For ion movement, for hyperpolarization, potassium is leaving the cell, which makes it more negative, and then for depolarization, sodium is going into the cell, which makes it more positive.
So the next pathway we'll talk about is the touch pathway. Its formal name is dorsal column medial lemniscal pathway. We're going to start with the sensory receptor cell, which in this case is going to be the passenial corpuscles receptor in the epidermis dorsal root axon. So this is a really long neuron, and it travels all the way to the medulla, where decussation occurs, and it crosses over in the brainstem. And after this, it goes to the thalamus and the VP nucleus, and then to the primary somatosensory cortex. The mechanoreceptor can be stimulated in 4 different ways: pressure, contact, vibration, or stretch. So here's what it would look like in a cellular aspect. So here's an example of an extracellular protein where a force would be applied and it would open the ion channel, letting in sodium and calcium, causing an action potential to occur and a depolarization within the cell. Same idea here, a stretching force opens the channel, sodium and potassium goes in, or here's cytoskeletal protein, same idea as all the other ones, just in different location. The difference between decussation for the visual process and decussation for the somatosensory pathway is that somatosensory pathway has full contralateral decussation, so it moves all the neurons from one side of the brainstem move to the other side of the brainstem, while in the visual only part of them do. That's why it's partial decussation. In the touch pathway, there are only 3 synapses, one between the passenial corpus receptors and the medulla and thalamus, and primary somatosensory cortex. So there's 4 different mechanoreceptor responses. There's rapid with a small field, rapid with a large field, slow with a small field, and slow with a large field. So basically, for the rapid section, there's an action potential as the pressure increases and like somebody's like touching somebody, and then as the touch continues, so like right now like this, there's no action potential, and then once you release again, there's an action potential to go back up. So same thing as here for the passenial corpus. And then for the slow adaptation, there's an action potential, many action potentials as you go down in the field, just like up here, but there continues to be action potentials as the pressure is constant, and then there's action potentials as they go back up again.